Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us for the 113th seminar, and I call it a University of Washington Day. I hope you enjoyed a lovely Valentine's Day yesterday, expressing love to your family, girlfriend, and boyfriends. Although we all love science, engineering, and technology, we should love people more than anything to make what we do more meaningful and significant. Without putting people above anything, what we do might lead to a dangerous or meaningless pursuit of knowledge or development. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor James Carothers. He is the interim chair of chemical engineering at the University of Washington in Seattle. More importantly, he is my academic uncle because his advisor was Jay Kisling, who is my academic grandpa. Well, we have known each other since the SynBYSS, the first synthetic biology center in the world, I guess since 2006, when that center actually started for the first time. James, thank you so much for your great contribution to the scientific community and also your amazing service as the interim department chair. I believe that is a huge job, and the virtual podium is all yours now. Thank you.
Well, thank you so much, Taesuk. It is a real pleasure to be here with everybody, and I'm going to make my remarks brief because, of course, we're here to hear from Sebastian and all the great work that he's been doing to combine machine learning and large-scale genomic analysis to help drive the design of new parts and systems. And so we're going to hear about that, and I'm excited to hear what he has to say. I'm going to tell you a little bit about how our group works very briefly, and then I'm going to tell you some big picture problems that we're interested in, and then I'll give you a hint of some of the technology solutions that we're applying to these problems. And so let me see if I can share my screen. And we'll get started. All right. So here at the University of Washington, our group engineers biology to produce chemicals and materials. This is the beautiful Mount Rainier, which is about 70 miles southeast of Seattle, on a nice sunny day. It isn't always sunny in Seattle, but sometimes it is. Our group, we are a group of about 18 or 20 postdocs, research scientists, and graduate students. They're all here. Our work benefits tremendously from some amazing collaborators, and in this deck, you will see little snippets of work that involve all of these people. But let's move on. We'll talk about some science. Oh, actually, one other thing. These aren't just dry names. We like to do a lot of things together. We go kayaking, we go skiing, we have cookouts. Again, here's that beautiful another sunny day in Seattle. We have to put those forward as much as we can, just to remind people not all the days look like this gray up here on the top left side. So we do have some fun too. All right, so what do we like to do? So we like to engineer biology to solve problems. And if we think about the kinds of challenges that are going to confront people who are starting their careers in the next 20 or 30 years, there's some really big ticket items here. And you could think of these as, well, you could be depressed by all these things, or you could think, well, these are actually opportunities for people to rethink the ways that we make all the things that make our life enriching. That is, enriching when we're not thinking about relationships that we have with other people, as Suk said at the outset. So there are still demands for renewable fuels, new and cheaper diagnostics, therapeutics, and drugs, higher performance chemicals and materials than the ones that we have now, strategies for low or no carbon energy and chemicals, new materials and strategies for information storage. I always like this number that IT is on track to consume about 20% of the world's energy by 2025, and that curve just keeps going up with the advent of new AI technologies. And then there's this idea here, that as with growing population and a desire to have higher quality living for more people, that we'd like new sources of crops, food and nutrient sources that place fewer demands on the environment. I think all of these things are really, these aren't all the important problems that people could tackle, but these are some of the ones that we think our work can have an impact on. And so what I'm going to do in the next couple of minutes is tell you about kind of fundamental technologies and approaches that could be applied to problems in this space. Generally speaking, how do we engineer biology to make the kinds of materials that we need to solve some of these problems? All right. So in general, we like this idea of developing biosynthesis platforms that can consume different kinds of feedstocks and then produce value-added chemicals, whether those are food, materials, medicine, personal care, other kinds of things that fuels, anything else that you might think of. Now, our group really combines these three approaches. We do large-scale metabolic control system design. So that is, how are we going to rewire metabolism to convert those feedstocks into the products that we're interested in? We spend a lot of effort, and this is work that we do with my good friend and collaborator, Jesse Zalatian at the University of Washington, to develop CRISPR gene regulation tools. And we've really pioneered a lot of the approaches for using CRISPR activation in particular to target specific genes and turn them on with timing and levels, expression levels that can be programmed. In the third bucket, we develop programmable aptamer biosensors as ways of reading out what these systems do. And a lot of this work recently has been spun out into a seed-funded startup company called Wayfinder. A group of alumni from our group are now leading that company. And so I haven't been talking about that recently as much in public, but these combined efforts, if we think of how to design systems, how to implement those designs, and how to analyze the results of those designs, those are really at the core of the work that we do. So now what I want to do is take, now that I've told you kind of what it is that we do, I want to take a step back, and then we'll kind of make this a little more specific. In principle, there are a vast array of chemicals and materials that you could make using biology. On this slide are just a set of metabolites that have industrial value. This is a nice Sung-Yip Lee review from a couple of years ago. If you haven't seen it, I highly recommend it. What's not always appreciated is that, at least here in the United States, bio-based chemical production is already a $160 billion a year industry, and we're on track for something like 25% of all chemical production to be bio-based in the next several years. So this isn't a niche activity, although it sounds like it sometimes when synthetic biologists talk about engineering metabolism. In fact, this is really a main economic driver, and there are, of course, really important aspects of scale up and downstream processing. Those shouldn't be underestimated. But the general challenge of, well, how do I engineer a microbe to make a lot of the molecule that I'm interested in, especially from a feedstock that I might be interested in? We can think about making molecules from glucose, but maybe even more exciting would be making this from waste materials, could even be carbon dioxide. And we can think about different ways of converting substrates that might be available to us into higher value compounds. There's a really vast space of possibilities here. So I think that's worth thinking about. Now, what I'm going to do in the next couple of slides is tell you why this is hard. And I'll do this in the following ways. The first is that when we introduce new functions to a microbe, there's already a pre-existing network. And we know that, genomes are really networks of networks. Evolution generates new cellular functions by forming new genomic links and regulation. And so really what's happening when we introduce new functions to this, we might perturb that network in ways that are really difficult to predict. And so in our group and with our collaborators, we usually prefer to take kind of a model and data-driven approach to doing this kind of engineering, where we think, okay, we're going to start with some genome scale model. This will give us some idea of where to start, but then we'd like to couple this with data-driven analysis. So now think ML model-based modeling. So that we can really take advantage of, well, this is an experiment that we did. This is what happened. Now can we use that information to drive the design of new systems? And I think that's really an important aspect of all the work that we're all going to be doing in the next several years. Next challenge. Let's use a specific example here. Now, so this is a molecule. We've been building systems to produce these modified aromatics in our group now for a while. It's industrial aromatics. There are something like 5 million tons of these produced every year. This particular molecule, 4-amino-cinnamic acid, 4-amino-styrene or p-aminostyrene, can be polymerized into materials useful for making photoresists, for making polymers, for PV panels, a bunch of other applications. You could think of these as engineered thermoplastics with tunable optical and mechanical properties. Now, in principle, you could make a molecule like this one in an organism like E. coli just by adding in 5 or 6 gene products. In practice, when you add pathways like this, you'll introduce control problems to the cell. So there are intermediates that are lost from the cell. There are other intermediates that will be toxic at relatively low concentrations. And so one of the things that we think a lot about in our lab are, well, all right, if a pathway like this one will kill an organism like E. coli, are there other microbes that might have physiological or metabolic capabilities that would make it better suited to be a platform for production? And in this case, we've been doing a lot of this work in a soil microbe called Pseudomonas fetida, which resists some of these toxic intermediates at 20, 40, 50 times more resistant to the intermediates in these pathways than E. coli. There are other ways you could build dynamic control systems to kind of, to program the timing and expression levels of the genes in these systems, but there are a lot of different strategies. But generally speaking, when we introduce new functions to a cell, we're going to create control problems that need to be solved. All right, one last one. That's a really, really big picture problem here. And that is that bacteria emit as much as 33% of any carbon feedstock that you give them as carbon dioxide. And if we think about all the molecules that an organism needs to persist, to grow, and evolve, there are efficient ways of producing those metabolites that go through, say, the tricarboxylic acid cycle. But each time you go around that cycle, you will emit carbon dioxide. So we're not so much concerned about the carbon footprint of the bioproduction systems that we engineer, but rather you could just think of this as, well, that means that 1/3 of the carbon or 1/3 of this hard-earned sugar or waste material that I'm giving to the cell, it's just going to be giving back in another waste product. And so the efficiency of any bio-based process could immediately be improved if you can reduce the amount of carbon that's wasted in some of these in its own central metabolism. So you can think about strategies like using cell-free systems, which is a big piece of work that we do in our lab. But you could also think of engineering carbon-conserving pathways, that is, pathways that more effectively convert the feedstocks that you give to the cell into the molecules that you're interested in. And if you can start to engineer bacteria to use carbon more efficiently, then this really changes the economics of a lot of the bioproduction that we do. And I think that's going to be an important area, not just for our lab, but for the industry at large. Okay, so the approach that we like to use, and it's kind of hinted here at the bottom of the slide, is to develop CRISPR-based gene regulation programs to rewrite bacterial metabolism. And our work in this area has been focused on really in 3 different levels. Here are just some examples of kind of recent work from the Carothers and Zalatan groups, so kind of combined efforts here. Developing new design rules, improving and generalizing synthetic gene regulation of bacteria, developing new tools, so creating CRISPR components for multi-gene expression programs. So we really want to rewire these metabolisms so that we can increase flux into the pathways that we're interested in and prevent the diversion of flux away from pathways that we've engineered. And then finally, a set of new applications. So building new multi-gene programs and non-model microbes and cell-free systems. Again, all aimed at this idea of, can we expand the number and range of platforms that we can use for bioproduction so that we can better tailor the bioproduction platform to the feedstock and to the molecule that we're trying to produce. All right, let's see. I've got actually just two more brief comments to make. So as we think about, over the course of the last several years, we've been really scaling up the number and the complexities of the systems that we can build. And so now we're working with a large number of collaborators to really think about how we can use transcriptional programs, uncover design rules for building really large CRISPR AI programs, implement those as dynamic CRISPR AI networks and to develop different carbon-conserving bioproduction platforms, with the idea being that if we can kind of dramatically increase the sizes and the scales and the complexities of systems that we can build, then we can really start to rewire those metabolisms for more efficient bioproduction. And so think on the order of dozens of genes and what possibilities, how many different things you could do if you could really start to easily, rapidly build and analyze systems that involve 25, 30 different gene targets at a time. All right. The last thought. Petrochemicals are becoming one of the largest drivers of global oil demand. It's one of those things that, we only use about 10% of a barrel of oil to make all of the other things that we use in modern life. But since this is also a major profit center in the oil and gas industry, we really need to figure out ways to get access to all of these kinds of chemicals and materials, drugs, clothes, even EV vehicles, if we're going to start to leave more of this oil in the ground and avoid the burning of it. And so I think, we like to think about this kind of as a grand challenge. Can we engineer biology to produce chemicals and high-performing materials that can displace petroleum and sequester carbon? And I think that I'll end there and just leave you with that thought as kind of a grand challenge for our age. All right.
Wonderful, James. That's all important problem. You're always giving the brilliant, amazing talk. So I thank you. And also, that's very interesting to me. You mentioned the IT will consume 20% of energy. And I believe you because my computer right now heated up. That means consuming a lot of electricity.
A lot of electricity.
And byproduct is the heat generation. And I hope my computer not blow up, but
that.
Anyway. Thank you.
My pleasure. Glad to be here.
Thank you. Okay, so now the main speaker of today with a longer introduction. Dr. Sebastian Castillo-Hair is a postdoctoral researcher in Georg Sieglich lab at the University of Washington, the same university as James. In his work, he used high-throughput experiments and deep learning method to study gene regulation and engineer novel functions in human cells. He obtained his PhD at Rice University, where he worked on optogenetics in Bacillus subtilis in Jeff Tabor's lab. Interestingly, Jeff, his advisor, is my academic brother who shared the same office with me at the Chris Voigt lab at UC San Francisco. Therefore, he is my academic nephew, as well as he is currently working in the same university as James, as I mentioned earlier, today's pioneer speaker. We can clearly see how all people are connected from today's speaker's relationship. Again, all starts with people and love about people, I believe. Sebastian, thank you so much for your time today. Please take it away. It's all yours now. Thank you.
Thank you. I was gonna make the academic uncle comment at some point if you didn't make it first. Um, yeah, thank you so much for having me here and for the very kind introduction. Yeah, I'm Sebastian Castillo-Hair. I'm currently a postdoc in the SELEC lab, uh, and I'm very excited to talk about my research topic today, which is to engineer human cell type specific enhancers using deep learning. And so I want to start with a. The slide is not moving forward. Okay, I want to start giving a little, just a very brief background on what enhancers are and how they regulate gene expression in eukaryotes. So in eukaryotes, you have the normal mechanism of gene regulation, which is you have a sequence called a promoter to which the RNA polymerase binds, and then it starts transcription from the start site of a gene. And then, but in addition to that, there's these distal elements called enhancers, which are located pretty far away from the promoter, sometimes several tens of or hundreds of kilobases away, that have open chromatin, that have binding sites for transcription factors, which can come in, bind to the enhancer, and because of the 3D proximity between the enhancer and promoter inside the nucleus, then these transcription factors are able to recruit RNA polymerase into the promoter, loaded with RNA polymerase, and therefore regulate gene expression from that specific promoter. Of course, enhancers, the interest that I have in enhancers is that they are central players in cell type specific expression. So as you might imagine, in multicellular organisms, you have that different cells from different organs and different tissues are running different genetic programs, are expressing different sets of genes at any given time to perform their diverse functions. And a simplified version of how this is regulated is something like this. So you have a cell type with a specific part of the genome. And this cell type is expressing transcription factor 1 and enhancer 1. Let's say that it has binding sites for transcription factor 1. So the transcription factor can bind here and regulate expression of its target gene and express the genes and the proteins that this specific cell needs to carry out its functions. On the other hand, in a different cell type, the same genome, the same DNA fragment will carry out a different function because transcription factor 2 is being expressed here. And so therefore it would induce expression from a different set of target genes and which would perform the functions that are specific to cell type 2. So this is a very simplified version of how cell type specific enhancer regulation works. But don't want to give the impression that this is a very simple mechanism. There's a lot of things that are not known about enhancers currently. So I'm taking a few of these problems from a recent review on Nature Structural Molecular Biology. For example, identifying and validating functional enhancers, just figuring out what parts of the genome are actually functional enhancers beyond like the 2 or 3 cancer cell lines that everybody uses is not a trivial problem. Then understanding the grammar, which is like how sequence determines which transcription factors are going to bind, which co-activators that are not necessarily transcription factors that are just in charge of remodeling chromatin or pre-opening chromatin, priming the chromatin, etc. There's a variety of things that have to happen in enhancers. It's not just binding of a transcription factor recruits RNA polymerase, and how the sequence of the enhancer determines that is, in the general case, not completely understood. Understanding transcription factor cooperation. As I mentioned, this is not cooperativity in the biochemical sense, strictly speaking. As I mentioned, some enhancers require binding of pioneer factors or factors that open up the chromatin, and that has to happen first before the actual signaling transcription factor comes in and actually recruits RNA polymerase. And of course, understanding the effect of mutations, which is very relevant for disease. A lot of the single nucleotide variants, or a lot of the variants in general that lead to genetic disease that are not located in coding proteins are thought to be located in enhancers and disrupt cell type specific expression. So of course, understanding what mutations do to enhancers is of crucial importance. But of course, there's also if we understood well enhancers and we were able to build enhancers that are specific to certain cell types, that of course a big biotechnological incentive for doing something like that. And in fact, people, it is a problem that people usually try to tackle to pick up cell type specific enhancers. So just to give an example of how it has been done in a recent study, this is a paper from the Allen Institute of Brain Science, also here in Seattle, Washington, where they try to pick a cell type specific enhancer from the genome using data from these fancy single cell assays, such as single nucleotide ATAC-seq and single nucleus ATAC-seq and single nucleus MC-seq. And then once they got that data, they had to spend a lot of work manually inspecting the transcription factor binding sites in the putative enhancers. And they were able to identify these pretty long stretches that contain candidate enhancers to get cell type specific on only one brain cell type. And then when they tested this they put this in viruses and they tested this in mice, and they found that about 30% or 7 out of 20 actually worked the way they intended. So this is not so long ago. This is from 2021. So pretty much the state of the art of designing or picking up cell type specific enhancers requires a lot of fancy assays, a lot of manual inspection, and even then the success rate is not as high as you would want. And this is all for one cell type. Of course, if you're not working in organisms, if you're just working in cell lines, and the work is a little easier, and you can actually test thousands and thousands of enhancers at the same time. So this is a study from 2016, which we're using actually as data to train our models. So what they did in this study was to take 450,000 sequences from open chromatin in 2 cell lines, a lot of which were, or you would think that there would be enhancers based on chromatin marks. And so they cloned this in library of reporter plasmids. So they located the enhancer upstream of a minimal promoter, and then they transfected this into these 2 cell lines, which is HepD2, which is a liver cell line, and K562, which is a lymphoblast cell line. And then they transfect the plasmid, they extract the RNA, and then they sequence the RNA, and then they can calculate the enhancer activity by dividing the counts that they get from RNA by plasmid counts. And if you look at what this data looks like, even though there's, for a lot of these enhancers, there's, due to chromatin marks, it would be expected for them to work as very specific enhancers. That's not exactly what we see for most of them. So this expression in HepD2 on the x-axis and K562 on the y-axis. And if a lot of these were cell type specific, you would expect, for example, the HepD2 selected candidate enhancers to be located in this corner over here, and the K562 data points to be located in this corner. But for the most part, they all, most of them have pretty low expression, and you are able to find a few that have higher expression, but not that many. And this is a pretty large screen with a pretty large and expensive oligo pool that produces the enhancers. So it is not like a huge success rate. But on the other hand, this is a very nice data set because it has a lot of points, a lot of measurements of relatively diverse sequence. So can we use this to train a model, and then to train a deep learning model, and then use a deep learning model to try to design the novel new specific enhancers that will work better than just taking the best sequences that you can get from the genome. And so this is exactly what we set out to do. And this is a modeling approach that was started by Chris Jean, grad student in our lab. And so what he started doing was to take a relatively simple convolutional neural network that receives 145 base pair candidate enhancer and tries to predict the activity in K562 and HepD2. We found that we could, with this modeling approach, we could get prediction accuracy that was on par with interreplica correlation. So this is kind of a noisy data set. So interreplica correlation wasn't that high, but even then we are able to train a model that matches interreplica correlation pretty well. And he tested a few different modeling approaches. So this is a bootstrap modeling approach in which you slice the data in different ways and just train different models to see, with different starting ways, to see if they can produce, if you got some variety in the predictions so they don't overfit. And this is an ensemble, which is basically just an average of all these models. And so then we try different, now that we have a good prediction model, we try different design techniques for this model. So for example, we could do stochastic search, which is to take a candidate random sequence and introduce point mutations and interrogate the model and see, basically ask the model, does this mutation improve expression in K562 compared to HEVG2, yes or no? And if it does, we keep that mutation and then we repeat that over and over again. Gradient descent, which is a slightly more principled way of optimizing sequences, will basically take the derivative of, for example, the difference between expression in HEVG2 and K562 with respect to the sequence. And there are some mathematical challenges with taking the derivative with respect to a discrete sequence, but we have a working method that can do that. And then a generative neural network approach in which that is probably you've heard something like this in the last few years. So we train a separate network that in response to random numbers can generate sequences that have desired features as predicted by this model. So with these three approaches, we generated 1,346 candidate enhancers. And then when we went to the lab to actually test them, we found that they were pretty well. So for example, I'm starting here with the original training data. And as a reminder, K562 specific expression should be in this corner, and HEVG2 specific expression should be more towards this corner. So we trained the model with this data, we generated new sequences, and when we measured the activity, we found that they actually improved over the original data in terms of getting you cell type specific enhancers. In particular, HEVG2 specific expression works pretty well. And in K562, it works better than the original data. We do get very specific enhancers with a higher success rate compared to the original data. But then what we can do to improve this even further is to retrain the model on these round one measurements and generate a new set of sequences. And you can see that in round two, that when we do that, when we retrain the model and design sequences again, we do get way more cell type specific expression compared to the other ones. You can see that the axis here goes a little higher, up to 6 compared to 4 and 2 in the original data set. So expression is higher, it's more specific. We improve our success rate for K562. So yeah, so by doing this, we've shown that we can take a data set, train a model, and generate sequences that extrapolate from that data set and generates and have performance that is even better than the training data, and that we can repeat this on an iterative cycle and end up getting sequences that are pretty cell type specific. And so one of the interesting things that we found when we tried to analyze why these sequences perform better than the genomic counterparts is that they tend to have very different grammar compared to natural sequences. And so we tried to do what's a pretty normal workflow, which is to take a database of transcription factor binding sites. This database is called JASPAR 2022. So it contains binding sites of all human transcription, or from a large set of transcription factors that have been characterized in vitro. And then we try to find enrichment in the sequences that were the most HEVG2 or the most K562 specific. And for the most part, the motifs that we're able to find make sense. HNF4A is a known liver type transcription factor. GATA1, TAL1 is pretty much what we expected in K562. But then after we did this alignment, we counted the number of motifs per sequence in R0, which is the original training data, R1, which is our first round of designs, and R2, which is the second round of designs. And we did find that the average number of motifs per sequence increases as we keep going on and on with this iterative cycle. So the deep learning model and the design algorithms are able to push the limits on how many regulatory elements they can cram into the same sequence space pretty effectively. And moreover, they tend to use a different set of transcription factor binding sites compared to natural sequences. So here in R0, I'm showing the distribution of transcription factor binding sites that were originally in the genomic sequences that we in the training dataset. And so with R1, you can see that there's already a shift in motif utilization. The motifs that were more enriched in the training dataset are not necessarily the ones that give the strongest activity, but the model and the method is able to pick up on the motifs that do result in the highest activity and use those to a greater extent compared to the natural sequences. And with R2, that bias is even more extreme. The model has been able to learn from the first round of designs what worked, what didn't work. For example, this motif SPKLF was used to a great extent in R1, but apparently it didn't do much, because then on the second round of design, the model apparently learned that it wasn't that effective compared to the other motifs and decided to not use it at all. So that is interesting to us, that these design sequences are able to extrapolate design sequences that are very different from the natural counterparts, and even then get better performance than the natural counterparts. One of the other fun things that we can do with this method is to try to design intermediate expression levels. So here on the x-axis, I'm plotting a target value of difference in expression in HEPG2 versus K562. And so sequences on the upper end would be the most HEPG2 specific, but we can intentionally target lower levels of cell type specificity. So we can design cells that are, or sequences that are a little less specific and a little less specific, and so on. And the same thing in the negative part of this plot, which is the more K562 specific. And then when we measure this in the lab, we find that their measured expression level is very well correlated with the target expression level, showing that we can design enhancers that maximize specificity, but also that tune expression in a very smooth way in the target cell line. So that's pretty nice. So now we've done this, we started to wonder how can we scale this up to more cells and more tissues, and try to see how we could push this into as close as possible to actual living tissues. And so one of the things that I mentioned at the beginning in my introductory slide is that these enhancer sequences usually are located in open chromatin. So you can see here the cartoons of the histones are absent in the enhancer. That's because the enhancer by its nature has to be open, has to be accessible to transcription factors to come in and recruit RNA polymerase. And so people have taken advantage of this fact for a long time. So the high throughput reporter assays that I mentioned a few slides ago, they're not that available in that many cell lines, and definitely not in actual tissues. But on the other hand, accessibility measurements of the entire genome are available for pretty much anything, at least in human, at least talking about human samples, in pretty much anything that you can think on cell lines, tissues, and these data sets produce a lot of measurements on a lot of sequences. There are single cell versions of these assays, and of course this data are so abundant that people have compiled them in databases. There's a database that I'm going to describe a little bit more in the next slide, of this DNA-seq assay, which is 3.6 million sequences for 400 and a little more cell types and tissues in humans. There's this resource called the Human Cell Atlas, which contains single cell measurements of different types of assays and different human samples. And so for specifically the assay that measures accessibility, I tried to look for how many samples they had yesterday while I was giving the finishing touches to this presentation. There's like 500 samples with measurements of 10,000 individual cells each. Some of them have more, some of them have fewer. And they're probably produced in the order of 100,000 sequences on average. And of course, these single cell samples are from complex tissues, so you can get very specific in like what cell type has this accessibility and so on. So if we could take advantage of this data, it would open up the gates to actually design enhancers on basically any cell type, and any cell type in a human organism, or even in animals in general. But then, of course, the big disadvantage is that accessibility is not necessarily enhancer activity. Sequence in the genome can be accessible for a number of reasons, and people, depending on the paper that you're reading, sometimes people assume that there's a one-to-one mapping of this, but if you read deep enough into the literature, there's plenty of examples in which accessibility doesn't lead to actual enhancer activity and so on. So it's not a perfect mapping, but there's a lot of data, so it is very tempting to try to design enhancers starting from accessibility data. So that's exactly what we try to do next. So we are collaborating with Walter Millman, who wrote a paper in 2020 on cataloging DNA's hypersensitive sites of a lot of human tissues and resulting in more than 3 million sequences. And all the accessibility information is well aligned, is well centered, is being curated pretty well. So just to give you an example of what this looks like. So for example, for hematopoietic cells, you can see that these part of the genome, this small segment of the genome is accessible in cells such as thymus, T cells, T helper cells, B cells, and not accessible in blood cancers, CD34, erythroid cells, and so on. And so there's a catalog of 3 million of these sequences in these 700 tissues. And just as a summary, what they were able to do in this paper is also to try to cluster this and catalog this and come up with like 16 of these clusters that are sort of like lineages that represent like accessibility programs that the human cells can do. And you can see things that probably make sense, like neural cells probably have slightly different accessibility programs compared to renal cells and so on. So we decided to test a very similar approach to what I described a few slides ago, but scale it up a lot. So we picked up 64 biosamples across all of these groups, and we decided to make a model, which is a CNN, but slightly larger than what I showed before, that could predict accessibility in these 64 biosamples. So for example, we still have HEP2 in this set, and you can see here that this model achieves pretty decent measurement prediction correlation. This is another example. This is a muscle cell line in which we also achieve pretty good prediction correlation. And if we compile all the prediction correlation for all 64 biosamples and plot it as a histogram, it kind of looks like this. So in general, we're able to achieve pretty good correlation in the order of 0.55, 0.6. And compared to which is already better than the previous model that we had only in two cell lines. So this is we have a working model, and so then we try to test in silico how to try to design sequences using the same approaches that we tested before, gradient descent and generative neural networks. And what we get out of this is something like this. So for example, for this retina cell line, which is where ERV1, we have accessibility that is predicted by the model to be really high in this cell line, but low everywhere else, maybe a little higher in all the other neural cell lines, which are SKNSH and fetal brain tissues and so on, which is to be expected because this is a neural biosample, and so it's going to be a little more similar to our neural biosamples. And so we did this for each one of the 64 biosamples in that are the outputs of our model, so we can design things that would be in theory specific to each one of these human samples. There's another sample that we sort of included in the model more as to give diversity to the model, not necessarily because we're going to test this. This is a fetal kidney sample, and of course we're not going to be able to test this in fetal kidneys, at least not anytime soon. But it is nice to see that we can design enhancers or sequences that will be accessible in theory here, and that would also have some intersection to other fetal kidney tissues, but are not accessible or not predicted to be accessible anywhere else. And so then we also did the motif analysis. So for example, the sequences designed by this method with HEP2 also have a higher motif density compared to the genomic sequences. And they also tend to use a very different grammar compared to genomic sequences. So here we can see the number of motifs per sequence of each one of these motifs. And you can see that the blue bars, which are the genomic distribution of motifs, are very different from the orange bars, which are the design the distribution of motifs in the design sequences. So this also use this feature of compressing grammar into a more tightly packed space. But then of course the question is whether these actually work in real life. So this is so this is data that we're still in the process of collecting. So this is going to be partial results right now, but I'm going to show you what it looks like when this works. So we have a set of, these are sequences designed for HEP2, and the y-axis is expression in that cell line. So include negative controls, which are sequences from the genome that are not expected to be accessible anywhere, and some random sequences. we included sequences that we just extracted from the genome by manually inspecting motifs and chromatin markers and so on. And some of these sequences work. As I mentioned, just picking up enhancers from the genome is not a trivial task, and it doesn't work all that well most of the time. So some of these worked. Most of them had a relatively low expression level. But then the sequences that we were able to design with deep learning methods have a pretty high expression in HEVI2. But then, of course, if we're designing cell type specific enhancers, you want them not only to have high expression, but to have low expression in other cell types. So we tested these sequences in all these cell lines, which include lymphoid, neural, retina, muscle, breast cancer, cervical samples. And we confirmed that expression of these enhancers designed for HEVI2 was low everywhere, but in HEVI2, showing that these sequences are very specific just for this cell line. And this is another example in which things work pretty well. This is Wary RB1, which is a retina cell line. And we can see that expression from the deep learning design sequences is really high compared to the controls. The genomic sequences are just barely higher compared to the negative controls, but the deep learning design sequences are much higher. And if you look at the other cell lines, there are like about 7-8 sequences out of 86 that had off-target expression. And that is something that we're in the process of looking into what happened there. But for the most part, the vast majority of these sequences are really good as cell type specific enhancers for this specific cell type. This is one case in which things work a little less cleanly. This is HeLa, a cervical cancer cell line. So we designed sequences to be specific for this cell line. And what we see is that, first of all, the two generative methods actually produce different performance. The generative neural network was able to perform better, but then there's also some off-target expression in HEVI2 over here. And in particular, the gradient descent method has only slightly higher expression in HeLa compared to HEVI2, the off-target, but the generative neural network did a lot better. It does have higher expression even compared to the liver cell line. And if we look at the individual enhancers, we can see that there are quite a few that have high expression here, and they're on the lower end of this box plot over here. And yeah, so in general, the approach seems to be working relatively well. And then for the cell lines in which things are working well, we can again try to tune the specificity of these. So here we design enhancers for different target accessibility levels, which is the x-axis over here, and then the y-axis measures gene expression. And again, we're trying to fine-tune. We're doing a weird thing because we're trying to fine-tune accessibility and then see if that leads to fine-tuning to some extent in gene expression. We can see that as a general trend, we are able to do that. We get a pretty nice correlation coefficient. And the same thing can be said on a different cell line, K562, which I didn't show on my previous box plots, but actually also work pretty well in terms of designing for maximal activity. But then we can also tune to a pretty good extent the activity in the target cell line with a relatively nice correlation coefficient. So this is where we're at. As I mentioned, this larger panel of data of enhancers in cell lines is still something that is being actively collected right now. So future directions is first to finish data collection of all this panel. And we're pretty excited to try to use motif analysis and deep learning interpretation methods to try to understand results, particularly things like this, where there's some off-target activity. We want to know what happened here, if these sequences truly have some regulatory elements that shouldn't lead to accessibility in HEVI2, but then are still leading to enhancer activity, or like some of these sequences in which there's off-target everywhere, and these are the same sequences in all of these. They're the same 7 problematic sequences. So we're very interested to try to figure out what's the failure modes, even when they're not happening all that much, or even when you can actually recover good cell type specific enhancers. We've been collaborating with Alex here at the University of Basel to try to bring this approach to zebrafish development. And also a part of my research that I haven't talked about is modeling 5' UTRs. We have a preprint on try to model the effect of 5' UTRs on translation during zebrafish development. We're excited to also try to use that model to design things that would lead to more or less protein expression earlier or later into development. I think a bit longer term, once we convince ourselves that this works, it will be very exciting to try to incorporate not only sequence information and not only have like different outputs for different cell types or cell lines, but also incorporate transcription factor expression explicitly in our modeling approach. That way we can, first of all, try to interpret which transcription factor is acting on which sequence element, but also try to extrapolate beyond cell types in the training data that we use for the model. Gene expression is something that is very frequently measured and for which there are a lot of data. RNA-seq is the most used sequencing assay, and single-cell RNA-seq is also the most used single-cell sequencing assay. So it would be very, very exciting and very nice to try to incorporate this into our models and see what we can design with that. And so with that, I'm gonna stop right there. I'd like to thank my lab, especially my PI, Igor Silek, and Chris Jean, who's a grad student that has been working with me in this project. Professor Walter Millman from the Altius Institute, who we've been collaborating for this accessibility part of the project. The Science Institute, which provided some funding at the beginning of my postdoc, and of course the University of Washington. And that's all. I'll be taking any questions now.
Amazing. Fantastic talk. Thank you. James, do you have any comment or question first?
I do. Sebastian, I wonder, do people, or is there a way to use gene ontology or, I don't know, ontology models to inform the design of these? I'm not even sure if it's design exactly, but to predict the performance of the enhancers.
So I guess what you will be getting at is something more like this. So one of the things that they try to do here is to pick up these sequences that are accessible in specific parts of the brain, specific cell types of the brain. But one of the criteria that people have traditionally used to select these enhancers is this close to a gene that has certain features, because it is thought that if this enhancer is relatively close to the promoter of a gene, then it maybe acts on that gene. And if it's known that this gene is cell type specific, or if it has like some ontology annotations or whatever, then they were going to pick that enhancer, and that will be, quote unquote, a strong candidate for being a cell type specific enhancer. One of the things that they find in this paper is that those things are not as straightforward as people make them to be. They very clearly point out that that enhancer proximity, for example, to a gene is not a good metric for whether the enhancer is going to be good or not. So the annotation of the gene in particular is not necessarily it may not be that informative because of the physical proximity, because physical proximity is not a good of a proxy of enhancer activity.
Okay.
So yeah, then the other way that you could probably do something like this, what I sort of alluded at the end of the talk, which is to try to incorporate information about the transcription factors, so not necessarily the genes that the enhancers regulate, but the genes that regulate the enhancers, and that's that'd be cool. There's, as I mentioned, there's a lot of gene expression data of transcription factors, and it is not known how you would use that to design enhancers at this point. At least I haven't seen anything like that, and also not models, at least based on deep learning. They're probably like biophysical models, but they make a lot of assumptions. That's also one way in which you can incorporate gene annotations. We'll look into that in the future for sure, because that's something that's very interesting to us.
Yeah. Yeah. Thanks.
Any other question, James?
No, that was actually top of my mind. I think, well, actually, I do have one other question. You mentioned, and maybe I didn't quite understand it, that the width of, let's see, how did you describe it? You said the natural enhancers versus the designed enhancers. I think maybe you can go back to that slide.
Yeah. Yes.
Yeah, that one. Could you explain that to me one more time? And I think, and then explain why it is that the naturally occurring ones seem to have so much more breadth. At least naively, I might even think that the opposite would be true, just because of uncertainty in the design or something. But yeah.
Yeah, so what's happening here is that so we take this database of motifs and we basically try to find occurrences of these transcription factor motifs in the sequences that were picked from the genome and the sequences that we design, just to get a sense of how many transcription factor motifs our algorithm is packing into the design sequences. And what we find is that the R0 here is a label that we've been using for the natural sequences for the training data. And so what we find is that the number of motifs per sequence seems to be higher in the design sequences compared to the natural sequences. So yeah, natural sequences have like 1 or 2, maybe 3 motifs per sequence, and then R1 sequences have a bit more, and R2 have way more motifs per sequence. So it seems to be because of the way we're designing sequences, the algorithm is packing more and more motifs into the same sequence, because the goal for us is to maximize cell type specific expression. So if the model thinks that it's going to get higher expression in the target cell line by packing more motifs, that's exactly what it's going to do. And moreover, I guess the point that I wanted to make with the last panel is that the motif utilization tends to be different. So the natural enhancers have some other motifs that, to be honest, is something that we really need to look into in detail. There are a lot of these that probably, like, they're called transcription factors because they have been found to bind to DNA in some in vitro assay, but a lot of these probably don't recruit RNA polymerase directly. They are dealing with chromatin remodeling, and because our assays are in plasmids, it is. I've tried to ask this to a lot of people, and nobody can give me a good answer of whether plasmids get chromatin remodeled or not. So it seems that it's not something that is very well known. So it might be that it just doesn't have chromatin by default, so you don't need a lot of these pioneer factors and things that remodel chromatin. And so for our task, the model decides that these are not useful, so it decides to not use them. And furthermore, for example, for the case of this TP53 gene, it is barely present in the training data, but it's so strong that our method decided to use it a lot on round 1. And once it saw that it worked pretty well, it used it even more on round 2. Same thing with HNF4A and so on. So yeah, that's more or less what's happening here.
Got it. Oh, that's cool. Okay, that's neat.
Okay, thank you. I have one question from audience. Maya asked, for deep learning interpretation, are you looking for explanation of your result or rules for rational design? And do you think the latter exist?
Well, both in general. It would be nice if we could just distill all of this into simple rules, and I think that's probably that would be probably a better goal that would satisfy more the people that are always looking for biophysical models. I think for this case of just deciding for 2 cell types, we've had some pretty clean conclusions in some cases that I have had time to describe very neatly, that are related to these motifs. Like more of this motif leads to good expression. More of this motif tends to repress expression, and that kind of stuff. For the larger panel of cell lines that I described at the end, I don't know if we will get something like that, but hopefully, especially if we incorporate, again, transcription factor expression, that would be pretty nice to do, but I guess we'll see.
Wonderful, thank you. I'm checking questions, so that is all we have. So now I also see 11, actually off 2, my clock. So let's close. There's a lot of data generation also, using the, deep learning. And one day I hope, somebody, including you, develop some algorithm. As long as we know the sequence, we know, how much expression happening in that sequence or something. So hopefully in the future. Okay, so let's close. Thank you all for joining and staying today. We'll meet again on February 22nd, Thursday, the same time, the same Zoom link. We'll have Dr. Ravi Basavappa from ARPA-H, a new funding agency focusing on innovative health-related projects, and Dr. Donghoon Lee at UT Southwestern. As usual, the following informal chat will occur without recording. Please stay here. If you are interested in chatting with us, I will promote you to panelists who could speak and show your handsome and pretty faces if you wish. And thanks. I stop recording. Just 1 second.